Hello, we're here at Tides USA, and today I'm speaking with Matteo Villain, VP of Peptides and Oligonucleotides at Veranova. Thanks so much for speaking with me today.
Thank you for the invitation.
I can't wait to learn a little bit more about you and your company, so let's jump right in. You spent over 25 years working at Bochem and other CDMOs across peptide API manufacturing. What made you decide it was the right time for a change, and why Veranova?
When Veranova, approximately 2 years ago, started looking into how we enter into the arena of CDMO for peptide, we look at the different options that were out there in terms of technology, and we decided to invest in developing LPPS chemistry. And we know that other technology to produce peptide, SPPS specifically, have a very high impact, while LPPS promised to be a more green and less impactful technique. I've been in this very large CDMO organization that are very good at delivering product again and again over a long time, commercial. But very often they are not set up to be innovative. They don't want to change their process that they have. And I believe it was a time for me to try to go in a place where I could use my knowledge to try to change the way we make peptide. And Veranova had this feeling that I could really express my interest, but also was a company that has an history of making active pharmaceutical ingredients. So it was not starting from zero, but they don't have a strong yet peptide environment where, therefore, I can be the one catalyzer of this change in the industry about how we make peptide for the future.
Well, it seems like you made a good choice. Now, what was it about Veranova, technically or culturally, that really caught your attention?
Veranova is a CDMO based in the US and in Europe that specialize in complex chemistry, and we manage very complex operation, and we have an experience in purification of some of this modality. Well, first of all, they are a company that had a long history of making small molecule, but also making special derivative, special complex compound, and peptide followed this group of difficult molecule to produce. So they were ideal for me. They are ideally set to use their knowledge in the small molecule to apply to the peptide. But also is a very young and active company. We have a very serious group of organic chemists, not peptide chemists, but real organic chemistry. And they really have both the chemical engineering aspect, meaning they are able to scale up process to very, very large volume, but also the chemical knowledge, scientific knowledge to help me put in place what I wanted to do. And this mixture of culture, I think, is what I was looking for.
Okay, and based on your experience, where do you see the peptide industry over the next several years?
Now, we are in a moment of peptide that I think you're aware of it, is very exciting because peptide came out of the corner of the less appreciated drug modality to become now a major modality that is of interest of all the major big pharma. The GLP-1 subset is only 150. The other one are all peptide that are used in many other indications, which include oncology, metabolic disease, immunodisease, and rare disease. There is definitely, we are in a trend of extremely high growth for peptide, both for the GLP-1 type of peptide used for obesity that demand very, very large volume, but also because peptides are now used in many, many other applications, also orally, that demand large volume. So it became a modality that was a niche with small volumes. And this is not only the use of the solvent, which is not green, but it's also all the organization around this plant that produce SPPS reactor has to be scaled in the same way. Just an example, I think, a molecule similar to terzipatide, which is a GLP-1, 100 kilo terzipatide require approximately 1.2 million kilogram or 1,200 tons of solvent for the synthesis and approximately 1.2 thousand tons of water for the purification. Everything is approximately 50-50. So the cost of making this material and the the environmental impact was not so important because of the small volume. As we go with the larger volume, that are demanded by this modality, we need to think about how to do it with a less important environmental impact. And then finally, there is tag-assisted liquid peptide synthesis, where you use a tag to replace the resin and to allow keeping the fragment as you grow them in solution. Now, how does the tag LPPS work? The tag is nothing else than a system of lipids that allow to maintain the growing protected peptide in solution as you grow the reaction. So you have a peptide that is growing on the tag represented here, and then you can add the amino acid to this solution, and using the same system of solvent and an activator, this will generate a acylation reaction at the end terminus of your growing chain. And then what we envision is adding the deprotection solution in the same mixture of solvent and reagent where we had the activated amino acid. Because you use a very low equivalent or excess of amino acid in this situation, the risk of deprotection and alkylation of the N-terminus is non-existing practically, and the excess amino acid that you have there most likely is consumed by the piperidine or the secondary amine that we have. And then we have two options at this point. We have developed precipitation, because these tags are very easy to precipitate with the product, so you can precipitate the product and wash it, or we can go to a aqueous wash and extraction. We have done both systems in our development, and we have found conditions to do this washing that allow to extract the reagent from the solution. And after the washes with the water, and we have conditions for the water, we do a separation of phase, and you go back at the beginning with having extended your amino acid of your peptide of one extra amino acid, and you can repeat the cycle. But interestingly, we can sample the reaction and in real time measure the extent of the operation of both the deprotection and the acylation reaction, which you cannot do in SPPS. And this allows you to now calculate exactly your excess amino acid, if you need more excess to drive the reaction to completion, and assure you 100% acylation of your growing chain and 100% deprotection. So this allows you to get higher quality crude. There is a technology called tag lipid LPPS, which means we do peptide not by fat peptide synthesis, by liquid synthesis. Now, in this situation, you can easily achieve higher throughput using very, very large equipment that is already used for other type of modality, and so there is no need to do investment. Now, with this specific technology that Veranova is implementing, but other companies are implementing, we are able to produce this drug for obesity or for other indications with a much reduced environmental impact. We can now implement green chemistry, solvents that are not dangerous for the environment. So ethically, I think it's the right things to do, not only to think at the volume that we need to do it, but how we do it with the minimal environmental impact. And this is the technology that we decided to pursue at Veranova.
That's very interesting. Now, peptide manufacturing has a reputation for being resource intensive. What's changing to make it more sustainable?
The sustainability impact, Veranova is committed to being a sustainable company that scales to the 100,000 liters reactors for small molecule. This new tag technology was developed in a way that reduced the consumption of solvent for every iteration of amino acid extension in the sequence. This already is important because you can reduce the solvent used for washes, you can concentrate the product, so you have now a less volume per gram of molecule that you produce. And this is already an important thing. And the other aspect which is important is with this technology compared to soy phase peptide synthesis, which is the way in the past most of drugs were made, allow you to monitor very carefully every single reaction with an exquisite precision so that you can titrate the reagent only to the level that they are necessary. And you can avoid wasting amino acids, which are very complex to produce and have an environmental impact. But additionally, at the end, when you finish making this peptide, there is always a step that is called purification, where you need to purify. Now, the material that is produced by this new technology, TAG LPPS, is purer compared to the material, has a higher purity compared to the material that we used to produce by SPPS, which means that the effort to purify are reduced. And since in this moment about 50% of the waste generated to make a peptide come from the purification, not only will we reduce the waste in the synthesis, we will also reduce the waste in purification. And by implementing in-process monitoring, you can obtain material with a higher purity before the final purification. So you streamline the purification. Is a win-win situation, both for the production and for the purification. So this is why these are more likely to have a reduced environmental impact.
Interesting, yeah. So related, how is Veranova thinking about sustainability in peptide production?
We have identified green solvent for this and established a toolbox of reagent, activator, and washing condition that we can plug in any sequence. We have initiated this program.
Yeah.
And to do this, we have developed a set of standardized chemistry to do it, which allow us now to focus on the selection of the green solvents, because once you have a system, you can do it in many different liquid solvents. But your choice from the beginning of which solvent you're going to use and which one you're not going to use define the impact. So Veranova has a commitment to measure, as we produce the material, the amount of waste that we generated and design the development with the objective to progressively reduce this waste, so making a greener process because you produce less waste, but also less hazardous waste. Because one of the things is water, I can use more water than some nasty solvent that pollute my rivers. The water comes back in the river, clean up, is still reutilizable. Some of these solvents, they have to be burned and incinerated, so the nasty one. And so if I can choose solvent, for example, ethanol, beside the fact that probably we prefer to drink it, but it's not allowed in the facility, but it will be a better solvent. We can obtain that from grain. So also thinking about where the solvents are generated, maybe solvents that are not generated from petroleum distillation are better. The solvents that instead are generated for fermentation of plants and things like that. So this is an overall holistic view of what we need to do to make it greener, and we are actively doing that.
Well, that's great. Well, thank you so much for speaking with me today.
Yeah, my pleasure.